All right, so we're on the last section here of chapter 2. This is the third part. What we have been looking at is also called the four macromolecules, and I identified that, I think, in the second section, but I didn't tell you what the four are. So let's talk about what the four are real quick. We have four macromolecules that the human body needs, and these are substances that we get energy from, and these are going to be those carbohydrates, the lipids, which are our fats, and now we're going to look at protein and nucleic acids. So that's the four: carbs, lipids, proteins, and nucleic acids. Those are really important things for the body, and this is how we get our energy sources and things that we need to build the components of our cells. So let me circle that little amine group here for you. That's NH2. That is your amine group. So it's NH2. Okay, they have a central carbon. You can see that little carbon there in the middle. So they have an amine group, the alpha or central carbon, and then they have a carboxylic acid group. That's that COOH on an end. Then they have the little lone hydrogen there at the bottom. And then this R here, this is a functional group. So everything but the R group in an amino acid is going to be the same. So you're going to have that alpha carbon in the middle, hydrogen underneath it, then you're going to have an amine group, the NH2 on one side and the COOH on the other side. And so they just have, it's kind of like a drill bit, an electric drill, and you have all the little different bits that you put in it to make it work, okay? And so these functional groups are the little different bits that have different jobs, okay? And so we take these amino acids and we put them together in different orders and in different amounts, and we make all of those 200 plus proteins that our body uses. So that's what's different. You can see here, this one on the left, that's the general structure here, okay? So this is the general structure. I'll erase all of that. Okay, general structure there. Then the next one is cysteine. It's got a sulfur bond. And then we have phenylalanine, and it's got that silic there on there, okay? That ring structure, that's that silic. Now we have four levels of protein structure. This can be kind of hard to understand. I do have a little simple paper model that I'm going to put online. I haven't decided yet if it's going to be in lecture and lab, but you're going to see that. And I also have a video because it's not hard to understand if you can put your hands on it and see it, okay? And so four levels of protein structure. So the primary structure is going to be this portion here at the top. And it's basically the order that the amino acids are put in. Whatever order you put any of those 20 amino acids in, and how many there are total, is what determines really everything about the protein, because the next levels are basically based on the order of those amino acids because they have the different functional groups, okay? So the primary structure is the order of amino acids. The secondary structure of a protein or a polypeptide, many peptides, okay, is going to be an alpha helix or a beta pleated sheet, okay? And so this section here, this is the alpha helix, and this is the beta pleated sheet here on the left, okay? This occurs due to those very important hydrogen bonds. Remember, I told you hydrogen bonds were important? And so you will have interactions between those functional groups on each of the amino acids that will cause bonding between those two amino acids, or three, or however many are involved in different places along this chain. That bonding is occurs between hydrogen and, remember, oxygen or nitrogen on another amino acid. So those functional groups can have bonding between them. And the order of those is kind of what determines if this is going to happen and where it's going to happen and what type. So you can have it form a coil shape, which is the alpha helix, or you can have it fold into almost like how you would fold a paper fan. Okay, and that's the beta pleated sheet. And that is based on hydrogen bonds occurring between hydrogen and oxygen on the functional group of these different amino acids, okay? You can have several of these structures in a peptide. So you could have maybe here, let's go with a different color. Maybe right here, you could have a beta-pleated sheet occur between some of those amino acids, and see each of these little links represents one amino acid. And then over here, you may have an alpha helix, and then you may have another alpha helix, and then maybe here it could fold, and you could have another beta-pleated sheet, and then you can have some strands that don't fold that way at all. Okay, and so this gives us a very diversified structure to all of the different proteins. And so you can have several of these which actually start to help form the overall shape. What is important about shape? What does shape determine? I've said structure determines function. Well, shape is the same thing as structure. So the shape of these proteins determines whether or not they're going to be able to function. So it's very important. Next, we have the tertiary structure, and this is where you're going to have some of these alpha helices and beta-pleated sheets have covalent bonds and some other intermolecular forces occur between the different parts of the polypeptide chain. So you're going to have those, and then you're going to have the alpha helix and beta-pleated sheets, and then they can bond as well. Okay, so they can bond, fold, and bond together as well, and that's going to give you an overall 3D, what we call globular shape, and it really does look like a big glob. And when you make your little paper model, you won't be able to do quaternary at home unless you make two, but when you make your paper model, you're going to see you do have just a big glob, all right? But it's a very specific glob that has a very specific purpose, so it's very important. So all proteins have up to tertiary structure. Not all of them have quaternary structure. Not all of them have quaternary structure. So what happens with quaternary structure is you take two or more of these 3D tertiary polypeptides and put them together. Okay, so you can have something like hemoglobin where there's actually 4 of these. You've got 2 of one type of globular protein there, and 2 of another type of globular protein there, giving it a quaternary structure, and then it's going to have a little iron complex here in the middle, and this is what makes hemoglobin. And this is why hemoglobin is able to be so efficient at carrying oxygen. It's all based on that structure. Okay, so to summarize the 4 levels of protein structure, you have primary, that's the order of amino acids. You have secondary, and that's where you have hydrogen bonds causing folding, which gives you alpha helix or beta-pleated sheets. And then tertiary structures is going to have more covalent bonding, and that's going to have interactions between those alpha helix, beta-pleated sheets and some of the straight portions. So it's going to have more interactions, cause it to fold even more, and that's going to be the tertiary structure. Now, there are some other forces and interactions that go in second and third, but I want us to focus on the main ones, and that's going to be hydrogen bonds for secondary and covalent bonds for tertiary. And then quaternary can happen sometimes if you have 2 or more of these tertiary 3D overall globular proteins come together and bond together as well, or have some forces of attraction between them as well to form that larger structure. So this is hemoglobin here. I wanted to show you kind of what it looks like. This is a diagram of what it looks like here. You can see I've got a 3D globular protein there, a 3D globular protein right there, a 3D globular protein here, and this one here, okay? And so you have 2 that we call alpha and 2 that we call beta. You can see in each of these that you have some alpha helices here. You can't see them too well, but there are some beta-pleated sheets, and then there are some straight strands there as well. So I wanted you to have a look at what that hemoglobin looks like in a molecular view. So that brings us back to the last of our 4 macromolecules. Let's review that list once again. What was the first one? If you said carbohydrates, you were right, and it really doesn't matter what order they go in. You just need to remember the 4. And the second? Okay, that's lipids. The third that we just talked about, proteins. And that brings us to our 4th one, which is nucleic acids. Do you remember what a nucleic acid is? Well, we have 2 very important nucleic acids, DNA and RNA, in our body, and they serve a very important function, okay? So nucleic acids in the form of genes carry all of the instructions that control a cell's activity. All of the plans to build proteins, all of the instructions that are needed. These are formed by a monomer that we call a nucleotide. And this here on your screen, that is a monomer, okay? That is a nucleotide, which is also a monomer, yes, but this is a nucleotide. Each nucleotide has a 5-carbon sugar, which is the portion here in the middle. That's going to be ribose for RNA, or deoxyribose for DNA. It has a phosphate group, which is on this end. Phosphate is PO4. And then it's going to have a nitrogenous or a nitrogen-containing base, and that is this one here. And this is, if you remember from high school biology for DNA, where you get your A's, T's, G's, and C's from. Adenine, thymine, guanine, and cytosine. And then in RNA, T, thymine is replaced with uracil, so it would be U. Okay, so those are the bases that we're talking about here. So that's the nucleotide there. These are going to form chains, and we call those poly. Remember, poly means many, polynucleotides, and those polynucleotides are going to be DNA, deoxyribonucleic acid, which is a double-stranded helical polynucleotide, and then we have RNA, which is going to be single-stranded polynucleotide. So here on the left, we have RNA. You can see that that is single-stranded. You have many of these nucleotides, so it's a polynucleotide. And then right here, we have an untwisted, non-helical, it's been taken out of the helix where you can see the structure better, polynucleotide of DNA, and those strands are held together by what type of bonds? Remember I told you it was really important, is the hydrogen bonds. So those are hydrogen bonds that occur between the bases.